Ti hei Māori ora. Te whare I tū nei, te whenua I te koto nei tino korua. E mihi nui ki te mana whenua o tēnei rohi. Waitaha kate mai mai, kai tahu, kina mauna whakahi, kina wai I tiri nei, kina rangatira huhua, tēnei te mihi, tēnei te mihi. Kina mate o te wā, rātou kuia, whetu rangatia, haere, haere, moi mai rā. Rātou kia rātou, tātou kia tātou. Ahurangi Bruce Russell, nā mihi nui ki a koe. He mihi hoki tēnei ki tō whānau me hoa. I nā rangatira, I nā ahurangi, I nā homahi, I nā tauera, I nā tangata katoa, tēnā koutou. Nō reira, e mihi nui ana ki a koe. Ki a koutou, sorry. Greetings to you all. I am Tony Ballantyne. I'm the Deputy Vice-Chancellor, External Engagement, and I have the great honour and pleasure of hosting this inaugural professorial lecture. A very warm welcome to all of you who have joined us here in our Castle Lecture Theatre this evening in person, and also greetings to those of you joining us online, either live or watching the recording at some point in the future. In my mihi, I acknowledged where we are, the location where we are gathered, mana whenua, who are obviously our treaty partner, acknowledge those who have passed before, and then returned to the room, greeting all of you, but especially greeting Professor Russell. I greeted your family and friends as well, and I especially want to acknowledge your partner, Noi, and your children, Mina and Matthew. I also pass on the warm regards and best wishes from Professor Helen Nicholson, our Acting Vice-Chancellor, who is unfortunately unable to join us this evening. Inaugural professorial lectures are important events. They are very special occasions. They embody and celebrate the endeavours that are at the heart of the university and its mission: the scholarly journey, the creation of new knowledge, and the transmission of knowledge created through research to students, to colleagues, and to the interested public. These are events with a very long tradition at the University of Otago. Right back to our foundation professors in the early 1870s, they gave inaugural professorial lectures, like us, to students, to their colleagues, but also to the interested public. So it is great that we have maintained this tradition, and it is fantastic that all of us have come together this evening to celebrate the work of Professor Russell. But it is also a celebration of the place of knowledge in making our community. I think that's enough from me. I'm very pleased to be handing over to Professor Day, who will offer a more formal introduction of Professor Russell. Nō reira, tēnā koutou, tēnā tātou katoa.
Tēnā koutou katoa, welcome and good evening everyone. It's great to see so many people here. I'm Catherine Day, and I'm the Deputy Dean of the School of Biomedical Sciences. It's my pleasure to be here on behalf of the Dean, Professor Lisa Mattison-Smith, who unfortunately couldn't be here but sends her regards. And so really it's an honour to introduce tonight's inaugural professorial lecture, or IPL, by Professor Bruce Russell. I'm going to get my words tangled. And so Bruce, as you all know, is from the Microbiology and Immunology department within the School of Biomedical Sciences. And it is really a delight. We are very lucky to have Bruce here in the Otago community. He hails from Queensland, which I think, again, many will know, where he completed his undergraduate studies at James Cook University in 1994. And then he went on to complete his PhD at the University of Queensland in 2001. Following his undergraduate studies, Bruce joined the Australian Defence Force, and he had postings to a range of interesting locations, and I think some of these were tropical. And so while I think he had an exciting time on a range of fronts, this time in the in the Australian Defence Force had a really big impact on Bruce's career, and he went on to complete his PhD in tropical diseases at the Australian Army Malaria Institute while in the Defence Force. So there was a significant period in the Defence Force that really has had a huge impact on Bruce's career. Following completion of his PhD in 1994, he then, 2001, he then went and continued as postdoctoral studies. So first continuing on at the Australian Army Malaria Institute, and then at the US National Academy of Sciences, but based in several of their different labs, and I think, again, spending time in fairly tropical places in Thailand and other places, and I'm sure he's going to tell us some of that in his story tonight. And throughout his time, really, Bruce was committed to advancing our understanding of malaria with really a huge focus on improving diagnosis and treatment. And this meant that he spent a huge amount of time in field laboratories, really working with patients and thinking about how to improve diagnosis and outcomes for these patients. And his research has had considerable impact, and he's received a number of awards during his time. And I was reviewing his CV before coming tonight, and one of the awards, he was designated as an alien of extraordinary significance by the US Army Surgeon General. So I think not many of us have had that designation. So maybe again he'll tell us tonight, we'll see. So postdoctoral study was followed by a period as a research fellow at the Howard Florey Institute, and he worked in the International Health Laboratory in Darwin for several years. And then from there he went to the National University of Singapore, where Bruce was an assistant professor and continued his work on malaria. So a long period of, I guess, building before he came to Otago in 2016. So from Singapore he came here, and really he's made a significant contribution in research and teaching since then, but as well as before. Bruce has a distinguished research portfolio, as we expect for someone promoted to professor, 120 publications and an H-index of 41. So by a lot of metrics, doing really well, but also having impact. So Bruce's work is highly collaborative, and he works with a range of partners in different centres, and I think this has been central to much of Bruce's success. And he's had a lot of really good papers since coming to Otago, and I note there are papers in Nature Microbiology and Nature Communications in recent years, very high impact journals, and it's a credit to Bruce's, I guess, the research calibre that he has been producing. All this research takes funding, and Bruce has had some success at getting funding from Gates Foundation, HRC, Marsden. These are all tough to get. The funding is tough to get, and it's, again, a good success. So research is not enough to be promoted to professor, and teaching is also something that I know Bruce is really passionate about and has had a huge impact, both mentoring students and taking leadership roles in courses and actually just delivering a lot of lectures. And so, of particular note, Bruce was the convener of Cells 191, one of the large HSFY papers from 2017 to 2021. This was both a time when there was curriculum review and COVID chaos. So I think it's a credit to Bruce that navigated this period, and so that shows leadership and commitment, which was really important. So Bruce continues to inspire students. He teaches at first year, and then he teaches at a range of other levels. And I note that Bruce has received a number of awards from the Otago Medical Student Association, really acknowledging the quality and I guess the impact that his teaching is having, because it's not always easy to get the medical students to think you're doing a good job. So again, that's impressive. So I really look forward to hearing Bruce's journey to Professor of parasitology in tonight's talk entitled Jungle Fever. So the stage is yours, Bruce. Congratulations.
Thank you very much. Yeah. It was about 11 o'clock at night, but I strolled on still with the people. Suddenly, on the left side of the road, there was a crackling amongst the bushes. All of us were alarmed. And in an instant, a tiger pounced upon the first of our party and carried him off in a twinkling of an eye. The rush of the animal and the crush of the poor victim's bones in his mouth and his last cry of distress, "Oh hi, oh hi," involuntarily echoed around all of us. It was over in 3 seconds. And then I, not knowing what had happened till I returned to my senses, found myself and my companions lying on the ground as if prepared to be devoured by our enemy, the sovereign of the jungle. I find my pen incapable of describing that dreadful moment. Our limbs stiffened, our power of speech ceased, and our hearts beat violently, and only a whisper of the "oh hi" of distress was heard from us. And it was in this state that we crawled on all fours and then ran for half an hour like an Arab horse till we reached a small village. After this, every one of us was attacked with a fever and attended with shivering, within which deplorable state we remained till morning. Although the author of this and his remaining companions escaped the warm jaws and 3-second death of a tiger, it would seem that all of them ended up with a fever which acted as a harbinger for probably something even more deadly than a tiger. This Malaysian jungle is handful to perhaps the few remaining tigers in Malaysia. And yet, as I sat here, breathing up the warm fumes and the humid geosmins coming up from the leaf matter around my feet, and listening to the cacophony of competing evening songs of birds and insects and monkeys, I had this ridiculous thought in my mind: what would happen if a tiger came out and ate me? Of course, a ridiculous thought. Yet, this tickling feeling was not paranoia, as I was certainly being observed. I was being observed by a legion of predators. And here in 2019, I was invited by these two incredible scientists, Professor Yi Ling and Indra, who's perhaps one of the most foremost medical entomologists in the world, Here we sat on our little crates in this jungle, one trouser leg rolled up, with a little torch and a suction tube, waiting for the imminent attack. Of the 3,000 mosquito species in the world, the Anopheles gambia mosquito is perhaps the deadliest. It carries the falciparum malaria. Only the female mosquitoes gorge themselves on blood. They need that for their eggs. The eggs are laid on clear water, and they hatch into larvae and then into pupa. It takes about a week going from egg to fully enclosed mosquito. And here's back to my leg in that Malaysian jungle. And this mosquito, hungry one, has been following me probably for an hour, focusing on the plume of carbon dioxide coming out of my mouth. Many people think when a mosquito bites you, it's like a hypodermic needle being pushed in and then the mosquito sucking hungrily like a thick shake up through a straw, but it's not the way it is. It's more like microsurgery, keyhole surgery. Here, the labium, which is this outer covering of the proboscis, covers up these incredible, nasty collection of instruments, 6 of them in fact. The outer 2, the maxillae, have razor sharp, and they vibrate at 15 hertz, cutting through the skin like butter. Then they used the mandibles to pull the skin apart and insert the other sharp instruments. The labrum. The labrum, this gutter-shaped microsurgery tool, draws the blood up. But not only that, it has sensors on it that can actually sense the red blood vessel, and it changes the vibration to 5 hertz to cut through painlessly into the blood. And the blood is then sent up to the mosquito, and it's concentrated. The water expelled out the back, and the red cells concentrated in. This is the important point. The center tool, the hypotharynx, injects the saliva. This saliva is a cocktail of antiseptic, anticoagulant, which allows the easy flow of blood up into the gut of the mosquito. But the actual injection of the saliva is the problem for us and for the mosquito. This infected Anopheles mosquito is currently injecting its saliva to help this blood come up through its mouth. But as you'll see, it's not only injected saliva, it's injecting malaria parasites, which we call sporozoites. These liver-seeking missiles are covered with a shaggy protein called CSP, which acts as a kind of countermeasure against the host immune system. Only a few of these sporozoites make it out and into the blood, maybe about 10. Most of them are sucked back. But of those 10, all of them will make it to the liver. Their target, the parenchymal cells. These liver parenchymal cells are where they want to go. But to get there, and to show their complete disregard for our immune system, they actually use our immune cells, like Kupffer cells, to actually burrow through as a gateway into our parenchymal cells. And there it'll work through our parenchymal cells, killing a few cells. No symptoms at this stage for us. But then it'll find its little home, and it will start to multiply. Oh, and what an amazing multiplication it is. From one copy of the parasite to 30,000 in a week. Again, at this stage, these are growing in your liver. You won't have any idea. There's no symptoms. Eventually, though, these 30,000 merozoites burst out of the liver cell and into the blood. Each one of those merozoites will never go back to the liver. It's a one-way street into our blood. because these merozoite parasites are actually after invading our red blood cells. And these are the juicy bags of hemoglobin that they target, full of protein that the parasite will use to multiply from 1 copy to about 30. Now, an interesting thing is that most malaria parasites, and certainly the deadly falciparum malaria parasite, isn't particularly fussy with what type of cell it invades. It'll invade any red blood cell at any stage. However, I've spent much of my career studying a not so deadly form of malaria, but an equally distressing one that causes incredible burden right throughout the world, particularly in the Asia-Pacific region. And that's Plasmodium vivax. Unlike falciparum and many of its kin, it doesn't like to, in fact, it won't invade normal, mature red cells. It's after the baby cells, the reticulocytes. In fact, this parasite will only invade very young reticulocytes. And the team of scientists that I've worked with, notably Professor Laurent Renier, Benoit Malloret, Rajesh, and Wei Hong, we've worked together on a number of important projects trying to work out how the malaria parasite, one, works out that it's going into a reticulocyte, or is this a reticulocyte or another cell? So we're looking at not only the receptors on the reticulocyte, which made us actually look really, really closely at reticulocytosis, because in fact this process from in the bone marrow where all of our red blood cells are produced, most of our red blood cells are produced as an adult, move right through this process, starting in the bone marrow and then being squeezed out into the sinusoids and out into the bloodstream. Interestingly, we found that Plasmodium vivax will only invade these very young reticulocytes. Importantly, we found that the very protein that our red cells use to bring iron into the red cells, which is the transferrin receptor, is actually the receptor that the parasite uses. Irrespective of what particular cell a malaria parasite enters, once it enters, that's when misery starts occurring. The parasite sheds itself of remaining proteins around the outside and creates a little bubble within inside the cell from which it can comfortably start chewing up the hemoglobin. It will then start to export a range of proteins to the cytoskeleton of our red blood cells, causing the red cell to become stiff and sticky. Most red blood cells are very, very deformable, flexible, and can move through tiny microcapillary beds. But the malaria parasite causes these red blood cells to become sticky and stiff. In this transmission scanning electron microscope, you can see an infected red blood cell of Plasmodium falciparum, the one that kills people. In fact, it kills someone every 30 seconds still. By the time of the end of this lecture, about 100 people have died through the infective bite containing this particular parasite. But becoming stiff and sticky for the parasite in exporting all those proteins and changing the red cell causes a problem for the parasite. Because every vertebrate has this organ called the spleen. And the spleen contains, amongst other parts of its anatomy, the white pulp, there is the red pulp. And the red pulp is a mechanical filter. This mechanical filter basically sorts all of our red blood cells. Every single one of us who has a spleen still will all have all of our red blood cells have to move from this side into the splenic sinusoid lumen. And to do so, it has to squeeze through these 1 or 2 micron gaps. A cell of 8 microns in size has to squeeze through these little slits. And it kind of filters and tests each individual red cell. Is it flexible? Is it good? And if it doesn't make the test, if it can't make it through here, it gets retained on the other side. These cells are then degraded, destroyed, and phagocytosed by a range of leukocytes that are found, white blood cells, our immune cells, that they're hungrily gobbling up anything that stays. And this is a problem for malaria parasites. If you're a stiff and sticky malaria parasite, you can't make it through there. You're going to be retained and concentrated here, where the white blood cells are. So throughout all my career and for most of the lectures, up until a couple of years ago, this is the story I've been telling students. The spleen was a parasite trap, parasite hell. We were wrong. Some incredible work recently led by Professor Nick Anstey of the Menzies School of Health Research in Darwin with his postdoc, Stephen Coe, and our incredible long-term collaborator, Enni Kanangalam, one of the most fabulous scientists I've ever worked with, and one of the few Papuan women who are doctors and academics working in the field today. And they found something incredible. They found that far from being parasite hell, a huge quantity, in fact over 90% of parasite biomass is actually being concentrated in our spleens. The parasites seem to be having a party in our spleens, which seems to fly against everything we thought we knew. How on earth can parasites survive in the red pulp? We found out how. Infected red cells, the malaria parasite, in fact, all species, malaria species, cover themselves with uninfected red blood cells. This is a process we call rosetting, malaria rosetting. And you can see the centrally infected red cell here, covered with uninfected red blood cells. My Brazilian colleagues, Fabio Costa and Letusa Ulbrook, and my Malaysian colleague, Wen Chao, and also my Singaporean colleague, Laurent Renier, we worked out that these rosetting parasites are impervious to phagocytosis. It protects them from phagocytosis. Malaria parasites cover themselves in a coat of your red blood cells to protect themselves from phagocytosis and clearance. Of course, up until now, there's been no fever. There's been no strain. It's only when that red cell bursts and releases all of the parasite proteins, parasite waste products, particularly hemozoin, that you will suddenly get that fever. That's when you get your fever, when all of those red blood cells synchronously burst open. And in fact, for many years, that is how scientists and doctors, for as long as we have known, written down medical case reports, have described malaria in terms of that periodicity between fevers. Some malaria parasites, most of them have a 48-hour period where it takes, after the invasion of the red blood cell to its bursting, is 2 days. That means you have a fever every 2 days. Some other parasites take 3 days. Some monkey parasites, like one that is currently going right throughout Malaysia and is killing lots of people very quickly, a zoonotic type of malaria called Plasmodium knowlesi, has a 1-day period. I've spent a lot of time trying to work out how red blood cells and how they've been affected, how the rheology of these infected red cells affect the pathology of malaria. And with Nick Anstey, when I was working with him in Darwin, and this amazing team of microbiologists, Kevin Tan, who's now the head of department for microbiology in NUS, and C.T. Lim and Li Young of bioengineering in Singapore, we developed these microfluidic chambers that actually mimic what's going on in the microfluidics. And we actually coated these microfluidic chambers in endothelial cells and receptors so that we could observe close hand. And not only that, we actually took these devices up to West Papua, into Timika, beside patients, up into the Thai-Myanmar border, beside patients, taking blood ex vivo and looking at it and examining it. Unfortunately, this particular isolate was from someone who passed away to malaria moments after we collected the sample. And it's not going to work. Anyway, what this video shows is red blood cells moving through rosetting in directly from a patient and literally clogging up and causing incredible downstream blockage, causing any kind of perfusion downstream of where the blockage occurs, and causing the patient, of course, if you have your brain infected with these stiff and sticky cells blocking circulation, the subsequent effects are horrific. This patient, who's given us, believe it or not, managed to give us permission to take this photo, came in. He was someone who'd recently arrived in Timika, who's a gold miner, panning gold. He'd never seen malaria in Java, but when he came to Timika, he was hit straight away. And within 2 weeks, he came to us in a coma. This is a touch and go period. You'll notice this bag, this from a catheter, urinary catheter. It's not blood, it's urine. These red blood cells that are burst and destroyed as they get ground up going through the capillary systems, causing terrible hematuria. My colleague, Sin Wen, who's an amazing ID doctor, incredible, in fact, very interesting story about Sin, is that he came to Dunedin before me. I think he worked on some mathematical modelling with Steve Duffel, and he also decided to go surfing here. But Sin had a very deep interest in looking at how we could perhaps use medications and other types of therapies to improve the outcome survival for people in coma. The best way is to give the person artesunate. Artesunate is an incredibly potent, and it is currently the mainstay of malaria treatment for severe malaria. Within 2 hours of treatment, the patient was responsive and was fine. And he quickly recovered, and we were talking with him very quickly afterwards. This is an incredible drug. There's only one problem. Since 2008, this indestructible drug that we thought we had is now resistance. And the resistance first started on the Cambodian-Thai border, and then it moved to the Myanmar-Thai border. One of my great heroes is Professor Francois Noston, who's an Oxford professor, but who's rarely enters the hallowed halls of Oxford because he spends all of his time living on the Thai-Myanmar border, working with the people, and doing his very best to work out how we can deal with drug-resistant malaria and to treat those who most need it. Aside from the drug resistance that we're starting to find creeping in throughout the Thai-Myanmar border, we're also finding resistance to other drugs, including chloroquine, which we use to treat vivax malaria. That's that malaria that invades reticulocytes that I was talking about earlier. These parasites are quite interesting in that they also invade the liver, but instead of taking 7 days to pop out, they're kind of like cats. They move when they want to. And these parasites in Plasmodium vivax will pop out at any time that we don't know when they will pop out. These dormant hypnozoites that we find in the liver of Plasmodium vivax pop out from time to time. And the problem with that is that means we get relapsing fever. Although it's not deadly, this incredible case study of a pregnant woman that we followed over a period of 200 days, she had over 9 reoccurrences of malaria. Despite numerous treatment protocols, she had it back again. Not only from relapsing, but also because of drug resistance. Because we found for the first time chloroquine resistance in this vivax parasite, which had never been found before. Much of this work, I should mention, is run by this incredible person, a pure angel. Rose Macready gives and gives and gives and gives. Another Oxford professor, very down-to-earth, self-effacing person who just wants to give to the people and do good work. She's only interested in doing work that helps women, particularly those who are pregnant with malaria. And in way of helping Rose, I'm going to give her a bit of an advertisement. She's recently started up a foundation. If you are in the business of giving some money occasionally to great foundations, I highly recommend Rose's foundation. She's an incredible person, and the work that she has done has literally saved, I would happy to say, tens of thousands of lives. Directly. So this business, not only are we seeing relapses, we're also seeing chloroquine resistance. Chloroquine resistance is important for vivax. Falciparum malaria, the deadly one, it's been resistant to chloroquine since the 1980s. We didn't think vivax, in fact, there was a lot of people out there who said, oh, it's impossible for vivax to become resistant to chloroquine. Well, it certainly did, and we found this particular woman had some serious problems with chloroquine. That's a real problem because chloroquine got a very long half-life. When you give someone chloroquine, it hangs around in the blood for many hours, many days. It's got a half-life of about 30 days, which means that when hypnozoites are reactivated within a 28-day period, it means that the chloroquine is still in the blood and it can still hit those parasites that are popping out again. So when we have chloroquine resistance to such a safe and relatively useful drug, particularly for treating pregnant women and children, we really have an issue. My early years in the army, that was one of the very first things I did, was working in Bougainville Island, trying to work out how bad the chloroquine resistance was. And that involved a lot of working with the local people who didn't particularly like me very much because I was Australian, and Australians weren't well loved in Bougainville. Kiwis were well loved, you'll be happy to know. But it was here that I had people such as Carl Reichmann, who developed the ex vivo drug sensitivity assay for looking at vivax. He invented it, and he was the director of the Army Malaria Institute. Jin Chen was pretty much my first boss in the Army Malaria Institute, an incredible scientist who's still at the appointment at the Australian Army and also with Queensland University. And of course, Rick Price. I've worked with Rick. After I left the Army, I was also working on chloroquine resistance in Papua, trying to work out what's going on. What we really need, though, is a drug that targets these dormant forms, these forms that pop out. If you go overseas, and let's say you decide that you would like to go and see the some orangutan in Borneo, and then you come back and you've been infected with Vivax, you could be driving your car one day and all of a sudden you'll start shaking and getting very sick. That's because these hypnozoites have just decided to pop out. This is a problem with Vivax malaria. And we need a drug that can eradicate those. The current treatment is basically 2 weeks being treated 3 days, 3 times a day with Primaquine. It's a terrible regime and it's poorly complied. No one ever wants to take that. So we want something better. And that's certainly what I was involved in my early time in my career. We started working with Mike Edstein, who's at this stage, he was a lieutenant colonel, and Major Dennis Kyle. This is at the Walter Reed, where I first started my work as a postdoc. But they wanted me to go out into the field. So I was still in the army at that time, so I went out with the Thai soldiers where we'd run the actual drug assays. And my job was there, taking blood off the Thai troops to make sure that they weren't getting sick from this new drug. And this new drug is called tafenoquine. Tafenoquine is a great drug in that we only have to give 1 dose instead of multiple doses over a couple of weeks. And it seems to work very well. The only problem is that it's hardly a panacea. It's about 60 to 70% effective at getting rid of these dormant forms. And the other problem that we have is that it's you can't give it to people who have severe G6PD deficiency. You'll remember that terrible picture of someone who had a coma due to falciparum malaria. Well, the fact that we have artesunate resistance has caused quite an emergency amongst the community. It hasn't become a World Health Organization emergency, unfortunately, but it's certainly amongst the malaria community, it certainly is. And we've decided that we had to produce as many new drugs as possible, particularly to kill falciparum malaria, those ones that stick in the brain, in the kidneys, and the liver. We needed something that would clear it as fast, and we did. We found the spirindolones, a totally new class. This work was directed by Thierry Degana, but his inspired chemist, Brian Jung, an amazing biologist, Pablo Biffini. This drug is now the fastest acting anti-malarial that we have. It affects basically the parasite's ability to maintain osmotic balance, and they basically pop. This is a healthy parasite up the top, and the parasites down below have been treated with the spirindolone NITD 609, which is currently in phase 2 trials. Now I come to the very important part of my talk. As Catherine mentioned, she noted that I have many collaborators. And I've mentioned quite a few of them as I've gone through my talk, but there is a special group of people who have always been there to back me up. And to the younger members of the audience, or those early in your research career, it's really important that you establish long-term mentors, people who are going to stick by you through thick and thin. And these are just a number of many. Laurent, amazing, just an amazing leader. George Sununu basically rewrote half of my papers. I'd send copies to George and he'd go through, and he has an amazing way of looking at things. Dominic Mazier and Carol Sibley have been people who I've never really worked much with, but every time I meet them, they want to know what I'm up to. We keep correspondence from time to time, and every now and then they're there helping me with references and things like that. But now I come to the most important collaborator that I've ever had and has been central to my career. The name hasn't been put on any of the other slides because I'd leave it to last. And she's sitting there and highly embarrassed. All of the work that I've mentioned on malaria, I've done with this collaborator. Almost everything I've done is been done with Dr. Rosarin Sawanarak. And I say Queen of Culture here. This is Rosarin, or Noi as she likes to be called, and this is Adeline Chua. I should say Queens of Culture because both of these women managed to culture some of the most difficult to work on malaria parasites. To do any work in malaria, you need to be able to culture the bug. And it's very difficult with malaria. And these individuals, Adeline, who's, by the way, an Otago PhD graduate of mine, I'm very proud to say, was also trained by Noi, as she trains all of the PhD students that come through my lab, and all of the masters, and all of the honours students too. So who is Noi? Who is Rosarin Sawanarak? Well, she's one of these people who is lucky enough to get their degree from a king, the great King Pumipon. And after her PhD, Noi produced some of the most important work on the pathophysiology of vivax malaria. Her work from her PhD was published in Infectious Diseases, and it's actually still like one of the classic texts that we look to look for how things work in this species of malaria. I thought it was a good idea that we actually formalize our collaboration and get married. Yes, that is me! Time hasn't been kind. Sorry. But Noi continued to decide to work with me through thick and thin. Some of her work has been amazing. This work, this figure, which appears in a science journal for the spirindolones, was work done by Noi. Figure 1, work done by her. She's world known for this particular type of work. Incredible. This was the linchpin of this paper. We were given powder, unmarked bags by Novartis. We had to go to the field and test it. And this work has been cited quite a bit because it's hopefully going to be the next big anti-malarial for treating. Noi has published over 50 papers, even some books on medicinal malaria. And it's not just all about promoting her own particular interests. As soon as she got here, she teamed up with two incredible women, Elaine Burgess and Rosalie Goldsworthy of Penguin Rescue. And since she's been here in Otago, Noi has been responsible for the molecular diagnosis, rapid molecular diagnosis and microscopic diagnosis of penguin malaria, which has killed tens of healthy breeding of penguins in Otago. So when I was told, when I was going to New Zealand, everyone said, oh, there's no malaria in New Zealand. Well, actually there is. There's avian malaria, and it's killing our little yellow-eyed penguins, kiwis, and a range of other native birds that have never seen malaria parasites before. I really have to thank, first of all, Noi, because I wouldn't be in New Zealand if it wasn't for Noi, because she was the one that handed me the Otago newspaper clipping and said, you've got to go for this. And I had one try at it and didn't get it. I tried again, and finally Clive let me through the door. Thank you, Clive. And also Vernon Ward. I remember the interview I had with Clive and Vernon, and thank you for taking a chance on me. I knew you were a bit worried about having a parasitologist here. Greg then took over as head of department, and I must say, Greg, thank you very much. You're an incredible leader, and I'm really, really sorry that you'll be heading back to my home hometown. It's not just a loss for Otago, it's a loss for New Zealand. Greg is one of the top 3 microbiologists in this country, and his move to QUT will be hard. And also Emily. Emily is just always there, directing things along, quietly working behind the desk. But I know that so much that happens goes through her hands, and she keeps the track going, and she keeps going despite negative comments and other things. Emily is a true professional. Thank you, Emily. I've made so many great friends and colleagues at Otago. James and I often pray together. Thank you, James, for your support. Alex, we often laugh together. And thank you for the cups of tea. You're a great buddy to have, and you've really been a great mentor while I've been here at the university. Muriel used to send me all of the interesting emails when I first were coming about what to wear and what not to wear in Remy, thank you very much, Remy, for joining my lab. Remy is incredible. He's originally from Yemen, and he's just really built an amazing research career. We've developed the only cryptosporidium culture lab in the Southern Hemisphere and one of 3 in the world. And it's all done by Remy and his amazing students. Megan, Warren, and Jen, I don't know what I'd do without you. You have saved my backside so many times. Thank you so much. Sorry for all of my mistakes. You guys are just amazing, and you really are at the heart of what this university is about, which is an incredible experience for students. Megan, you put up with all of the biosecurity stuff. Luckily, I've got Noi, who's managed to keep things ordered. If I didn't have Noi, I know that Megan would want to kill me within. And just a few other people I'd like to mention. Tin, thank you very much. Tin and I have been to Myanmar a few times to look at a few projects up there. An incredible colleague. I also want to thank Alan Clark and Chris Harry. They're no longer with us anymore. But when I first came to the department, these guys, I used to have tea in the morning, coffee in the morning, and little breakfast club. And they're just amazing people. You know, their job description was like this, but what they did was this. Because they loved us. They thought we were family, and so they would do so much more than what was on the job description. I'm sorry to see them go after SSR or whatever it's called, that name which we shouldn't mention. And finally, Alan Hughes, who stepped into the very large shoes of Alan. Things have been tough sometimes here, and before I arrived at this university, I have a bookcase full of Christopher Hitchens and Dawkins. I was a big disciple of theirs. Came to New Zealand and become a Christian. So for those of you who are rolling your eyes now saying, oh no, I'll blame you guys, because I was a self-respecting atheist before I came to this beautiful country. But certainly these people have been there at some of the tough times. We've lost people in our department, and these incredible people have been there. Rose Scott runs a family service for that I bring my children to first Sunday of every month at the Trinity Anglican Church, and the last service we had was a blessing of the animals. So all of the congregation brought their animals, dogs, and we brought our orphan lambs. And this is my son Matthew getting just the beginning of this month having our lamb blessed. And I tell you what, I haven't felt like blessing them when I'm getting up at 6 o'clock in the morning to feed them. I feel like blessing them with, no, I won't say that. So thank you very much. I'm very, very glad to be here, and thank you all very much for coming along to my talk. Otago University has 2 things that really spoke to me. One is it had the lifestyle that I really desired when I moved from Singapore. And that was the thing that really attracted me to this place, was the incredible lifestyle and the incredible New Zealand people that I met. The second thing is, I will say this without any shyness at all, this university treats students like no other university I've ever been associated with. The level of care given to undergraduate and postgraduate students at this particular university is astounding. And I am not shy at all to say that, because it's true. And anyone who works or teaches knows this to be the truth. So thank you very much, and blessings to all of you. Kia ora.
Thank you, Bruce. Now I get to say a few words, so you can sit down. Tēnā koutou katoa and warm Pacific greetings to you all. My name is Greg Cook and I'm the Head of Department of Microbiology and Immunology. Thank you, Bruce, for your IPL tonight. Every time I hear you speak, I've heard you speak a number of times, but I just get inspired. Your praise of others is just remarkable. You acknowledge everybody in the room. You are truly gifted. If David Attenborough ever needs a replacement, I suggest that you might have a role to play. So thank you, Bruce. Perfectly pitched. So tonight we heard all about Bruce's world-leading research on malaria. Sorry it took so long. I think we made you professor in 2020, and here we are in 2023, but a few things got in the way. Evident in your talk tonight was your total passion and commitment to basically studying malaria, development, diagnostics, vaccine testing in endemic countries like Malaysia. We should have huge admiration for researchers like you, Bruce, working at the coalface of infectious disease research, very simple laboratories. And, I'm not sure I could sit in the jungle at night being bitten by mosquitoes and waiting for a tiger to eat me. So your considerable bravery, and I'm pretty sure, Matthew, you probably weren't doing much on your fortnight, because I think, Bruce really held our attention for the whole 45 minutes. The panoramic scope of your work and recognition of your achievements by the scientific community has been demonstrated by your talk tonight. Many outstanding contributions to the field. I saw a number of the papers coming up, Nature, Science, Blood, PNAS. You are very humble. You are the person that has also driven this research. I admire you that you pay respect to all these other researchers, which is fantastic, but you are also a major instigator of this research. And what's been really pleasing, as you said, we were worried when you came here about whether you'd be able to do malaria research here, but I think you've proven us all wrong. So congratulations, Bruce. Something that Bruce didn't talk about was that he was able to pivot his research to other parasites, and I think over the last few weeks with the Cryptosporidium outbreak in Queenstown, Bruce has featured on the TV and the radio, and I think this is a very exciting phase, and I just wish the DVCR was here. I wish the Vice-Chancellor was here, because I think we need to support this research. We can't let this research go under because we can't get funding, right? Once we lose that capability to do this work, we'll never get it back. So I spoke to the Commissioner today. The Minister is going to be contacting the university, so hopefully we can get some momentum behind this. Not only is Bruce a world-class researcher, as we saw tonight, he is an absolute tour de force in our teaching program. And as you saw tonight, in any oration, any talk, any lecture I've given, passion, excitement, humour, and legendary Aussie storytelling, right? So thank you, Bruce. You're amazing. My daughter did first year science, and she said you were the best lecturer by a mile. Bruce teaches at the peak of first year health anxiety, so he comes in right at the depth there where anxiety is through the roof, and I can tell you that Bruce has a really calming influence on these students. These are 2,000 students, they need a lot of pastoral care, and Bruce really is the calming influence with that audience. I think it helps that you also tell them what's going to be in the exam, so this is probably something that I need to do more of. Yeah, I know. So, I want to thank your amazing family. We saw tonight that Noi, in her own right, is a world-class researcher, and Bruce, thank you for acknowledging that. So thank you, Matthew and Mina, for sharing Bruce with us. I mean, I'm sure some of those stories with the tigers must scare you when he's away. But yeah, an amazing researcher, and so thank you, Bruce. So hopefully this evening was a fitting celebration to you to recognise what an amazing academic Bruce is. Congratulations, Bruce, and thanks for representing the department and the university with such distinction, both on the local, national and international stage. I'm going to miss you, but I'm sure you'll come and visit me in Queensland. So thank you, everybody. I have a small gift.
Yeah, thanks, mate. Yeah, cheers. Yeah, thank you. Yeah, thanks.
Goodness. Never been in such a dark lecture theatre. Kia ora koutou, I'm Trish Priest. I'm the Acting Pro-Vice-Chancellor of Health Sciences, which is the part of the university in which the Biomedical Sciences School and then the Department of Microbiology and Immunology sits. I have a very short task, you'll be pleased to know, but a very pleasant one. It's always, one of the great things about being in my role is that I come to lots of these inaugural professorial lectures, and they're always interesting. They're not always as dramatic as this one, and that was really engaging and fantastic. Thank you so much. I've never been to any of your student lectures, but I can imagine that they're equally engaging. It's always great to learn something more about the work that people do here and about the particular person who's a colleague in health sciences, and so this has been a great one, and it's lived up to all my hopes of going to a great IPL. So thank you so much, Bruce, and many, many congratulations. My task is really just to ask you to, when I've finished, give Bruce one more round of applause and then come and join him in the staff club for refreshments and continue the celebrations. So once again, thank you, Bruce, congratulations, and thank you all for coming.